Okay, good day. Today we are going to be talking about the pattern of inheritance. I mentioned this from our last lecture. Now, autosomal dominant inheritance occur when only one mutant allele is sufficient to produce disease. Now, the condition typically appears in every generation, and males and females are equally affected. An example of this is Huntington disease. Autosomal recessive inheritance requires two mutant alleles for disease to be expressed. Okay, now parents are usually asymptomatic carriers. Now, this pattern is more common in consanguineous marriages. An example is sickle cell disease. Now, in malaria epidemic region, um, in malaria epidemic region, the carrier state provides partial protection against malaria, okay, explaining its high frequency in certain population. Now, X-linked recessive inheritance predominantly affects male because they have only one X chromosome. All right, listen. There is no male to male transmission in this aspect. An example is hemophilia A. Okay, now let's look at mitochondrial inheritance. This one is transmitted exclusively through the mother because mitochondria are inherited from the ovum. If you remember, now disorders from this often affect high energy tissue such as the brain and muscle. Okay, um, now uh, 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 what are the molecular basis of genetic disease? Because genetic disease they have a molecular basis. Uh, uh, they may arise through several mechanisms. Loss of function uh, mutation result in reduced or absent protein activity, often due to enzyme deficiency. Gain, gain of function mutation, this one is produced, it produce abnormality or abnormal proteins with new or exaggerated activity. Okay, now uh, there's another one called the dominant negative mutation. This one occurs when an abnormal protein interferes with the function of the normal protein. Now, haploinsufficiency occurs when one functional copy of a gene is insufficient to maintain the normal function. For example, in sickle cell disease, a single nucleotide, for example, a single nucleotide substitution lead to the replacement of glutamic acid with valine in the beta globin chain. This molecular alteration changes hemoglobin structure, resulting in hemolysis, okay, vaso-occlusion, ischemia, and multiple organ damage. Now, genetic testing in clinical practice. Uh, Genetic testing plays very important uh, important role. Okay, it's increasingly important in medicine. Actually, it may be diagnostic, it might be preventative, predictive. Sorry, it might be used for career screening, even prenatal diagnosis or newborn or screening. It can all be used for this. Now, in oncology, for example, identification of specific gene mutation helps to guide targeted therapy. For example, mutation in um, mutation in BRCA gene significantly increase breast and ovarian cancer. Okay, ovarian cancer risk. Now, pharmacogenomic studies have genetic variation. Uh, 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 I mean, pharmacogenomic studies of how genetic variation affects drug response, allowing personalized drug selection and dosage adjustment has also been utilized. Now let's talk about gene environment interaction. Gene alone do not determine disease. Okay, if you remember in my other video I was telling you about the role of environmental factors. So yes, environmental factors such as nutrition, infection, toxins, stress, and even lifestyle significantly modify genetic risk. For instance, um, an individual with genetic susceptibility to type 2 diabetes may never develop the disease without environmental triggers such as obesity or sedentary lifestyle. Okay, now this interaction actually explains why identical twins may not always develop the same disease. All right, now let's talk about epigenetics. Um, Epigenetics actually refers to heritable changes in gene expression that occur without changes in the DNA sequence itself. All right. Mechanism of this include DNA methylation, histone modification, and non-coding RNAs. Now, these processes regulate whether genes are turned on or off. So epigenetic alterations are actually implicated in cancer, developmental disease, and possibly transgenerational disease transmission. Now, a part of genetics that is actually very important is the ethical, legal, and of course social consideration of it. Now, genetic information raises important ethical issues, including confidentiality, potential discrimination, reproductive decision making, and gene editing. And also, advances in genome editing technology, such as the CRISPR, have opened new therapeutic possibility, but also raise complex ethical debates regarding, germline modification. So this is actually very important, the ethical, you know, and the legal part of it. Now, the clinical importance of human genetics, like I said, is very central to modern medicine. Okay, um, it underpins oncology, reproductive medicine, neurobiology, cardiology, and the rest. Even my area, endocrinology and infectious disease research. Now, so understanding genetic principle actually enables, you know, clinicians to diagnose inherited condition and assess disease risk, counsel family, and practice precision medicine. So, human genetics is not just limited to rare inherited disorders, okay? It's something you need to understand. It is the biological foundation upon which health, disease susceptibility, and therapeutic response are built. So, as future scientists, clinicians, mastering the principle of human genetics, we actually empower you to move from symptom based medicine towards predictive, preventive, and personalized care. Okay, so thank you very much. We'll meet in our next lecture video. Thank you.